As you mentioned, my name is Pedram Ramin. I'm a senior researcher, and I'm talking about SmartSens. This is a spin out from Technical University of Denmark, DTU. And like other PAT, I think technologies, we want to accelerate the shift to digital and sustainable bioprocessing. Just to state some of the obvious and the problems that you're all familiar with in food and pharma industries, where we measure offline and samplings to measure small molecules, metabolized products, taking a lot of time, and we're missing a lot of information. I think our estimate is that the 50 to 70% of the time is wasted actually on these offline practices. We get the results with big delays, and that also costs a lot, 10 to 30 billion euro per year from our own estimates because of delays and yield loss and process development. So we have developed in the past, I would say, 5 years, novel bio-free electrochemical sensor technology. So these are basically similar to biosensors that you all probably familiar with, but without the bio part. So we combine different elements from electrochemistry, nanotechnology, and polymer science to make the detections of these small molecules. And we can measure glucose, lactate, and ammonium real-time inline in the solution inside the bioreactor. So the sensor device, you can see it here, we have produced it, and then this integrates sensor elements. So that sensor elements basically integrates with the sensor device, and it needs to be replaced after each reactor run, and you can get the signal out. The way it works is based on amperometry. When we apply voltage, and these small molecules or analytes would interact with the working electrodes, and they go under reductions or oxidations. For example, if they reduced, and then they also release electrons, and that electron can be amplified, and then the signal can be correlated with the concentration of those small analytes in the solution. So amperometry has been around for many years. The way it is special we do it is we don't have any biological elements to mediate the electron transfer, like enzymes or DNA or any biological elements. So it's a direct electron transfer. We call it fourth generation of electrochemistry. We also do that by applying a specific, I would say, very sensitive materials with 3D structures made in the clean room environments to enhance the surface area for the reactions and detection. This is a sensor anatomy, multiple layers. We have the electrode base, is the working electrodes with this nano pillar structure, 3D structures. We have the coating layer of applying different chemical molecules, so you can capture the analytes and interact with it, like glucose in this case. And then we have 2 layers of protection to block basically these larger molecules and debris inside the ferments. Just to show you some of the results for our 3 measurements of glucose, lactate, and ammonium, show the dynamic range. It's very linear, the dynamic range. So you see the current density versus the concentration of the analytes. For glucose, we can measure from 0.1 to 120 millimolar, a very straight line. Similar to lactate, 0.01 millimolar, very low concentration, all the way to 90. And for ammonium, it's actually up to 200 millimolar. So very broad dynamic range, and this is actually exceptional. And the linearity is also very high. Something about the sterilizations that we have investigated. This is with gamma radiation. The figure on the left, which is with 25 kilogray. So before sterilization and after sterilization with gamma radiation, you can see actually the slope of the calibration line actually doesn't change. Similar to the 50 kilogray on the right side when we apply this gamma radiation. We also have tested it actually for steam sterilizations. We could at the moment go up to 90 degrees steam, but after that, because we apply different coating materials, this coating actually melts. It's not very useful at the moment. We are working on different coating material that can withstand also temperature up to 140 degrees. But we can for sure sterilize the sensor with gamma radiation. About the stability of the sensor, this is actually the very big advantage of having bio-free sensors. So in operation, we tested it. They can last for 25 days. Of course, the response drops. You can actually try it more if you want to. And the shelf life stability, we tested it after 6 months. So basically, you can leave the sensor in room temperature in the lab environment, and then you come back and then use it again. This is very important, for example, for single-use bioreactors when you have you may need a long shelf life stability. Just to show you some of the results, this is for online measurements where we have integrated the sensors into flow-through system to measure glucose in yeast fermentation. So you can see during a 10 hours fermentation run, we can match those HPLC measurements that are done every hour with our real-time measurements, this online system with the platform. And then we also did a test to check the response of the sensor by spiking with glucose. Similar for offline testing with the mammalian cell cultivation, when we compared our sensor, SmartSens, with the Bioprofile Flex 2, it's also bio-free. And then we can see actually for all the analytes of lactate, ammonium, glucose, which is all important for CHO cells, for example, we can also find a very good match. The application of the sensor is very broad. We can apply this one in various systems because it is the sensor is highly flexible in design. It can be miniaturized and it can be integrated in different systems. We are working in different systems now, glass, stainless steel, single use, especially because of this high stability. I think this is very, interesting application there. But also we are trying to look into well plates and also microfluidics because it can be, as I mentioned, miniaturized. The team behind is myself, is Alia. You may know her. She's the main inventor of the technology. And we have Shova and Ella, also electrochemist and analytical chemist. We have Chris as our advisor. You may also know Tor Rasmussen. He was the former CEO of Friesens, and also Stefan is the former CEO of C6 Sensor. So we are trying to gain people or gather people with a lot of experience in sensor technologies. And we are looking for collaborators in also developments, not only testing, because we are also trying to actually develop these biofouling issues. We also have them, and we're trying to develop better coating materials and better membranes to protect the sensor. And we also thank you all the foundations actually that gave us the funding in the past 3 years. And please follow us. Thank you.